Hello, this video will cover sample variants, how to find them within the system, and how to utilize them. When you first launch the software, you're presented with multiple tabs. We will be using the SNPs tab for this video. The first thing we'll want to do is select the organism that we're working with. So for this scenario, we'll select Arabidopsis. When you select this organism, you're presented with all the samples that are currently in the system. So in this scenario, there's 1,308 samples. Each sample has a name, could have a description, has the number of SNPs that have been annotated with it, and also there's a list of different trait types associated with these samples. For this scenario, we'll use seed dormancy, which I've selected here and has populated a column here. If we sort by this sample set, we now see which samples actually have this trait. We'll select a number of those by using the Shift and the Control and the left mouse button. So we'll select 20 samples and we'll add those to Persephone. Now you'll see a representation of those samples here with the names across the top, and these are all color-coded based on the reference genotype, as seen here with the radial button. All blue is the same allele as the reference genome, all red is the alternate allele to the reference genome. You can bring up multiple chromosomes by holding down the Control button and clicking the chromosome. You'll see that there's a distance here, and so the samples are ordered based on the distance from the reference genome. We'll select chromosome 1, we'll left click on that, and that will bring us to the maps view, which now allows you to see the variants with the genes and other tracks that you have available. If we zoom in to the region, we'll see the genes, we'll see variants across the different genotypes, and as we move across these genotypes, you'll see the little pop-up balloon tell you what genotype you're on. And also if you hit the shift bar, it'll also tell you at the top of the screen. If you use the Control button and the left mouse button, you can select a region, and then if you left click on the selected region, it will now bring up the horizontal view. In the horizontal view, you can quickly use your mouse wheel and go all the way down into the single nucleotide view if you would like. So within this view, you have your samples, you have your SNPs that are within those regions, and you have your genes that have been annotated by different mechanisms. When you scroll over a given gene in the SNP section, you'll see a gray box. When the box is grayed, you can right click and get additional options. For this video, we'll just generate translation for gene. When you generate a translation, each sample is translated based on the annotation that's selected and based on the SNPs that are within that sample. If you see a flag, that means a SNP, which is an A in this sample, and the reference is to T, has produced a different amino acid sequence. And you can see that and zoom to that in multiple areas. And you can do that for any translation. So if I select this translation, they change, and now we have mutations here, or I can select this translation, and now I have mutations here. All done in real time, and so immediately you can understand what effect SNPs are having on the gene that you're interested in and the different translations of that gene. There are many other things that you can do with this page if you go to these different options, and we'll cover those in other videos to come. So I hope this helps you getting started with SNPs, selecting samples, and then evaluating those on a smaller region of a selected genome. Thanks.